So good afternoon, everyone. Today we have Ivan Capo. He's from the University Clinical Center of Vojvodina, which is placed in located in Novi Sad. Is the second largest city in Serbia, right, Ivan?
Yes, that's right.
And he's gonna talk about the role of histopathology in the assessment of animal models in DILI prediction. So thank you very much for giving us this this talk. And you can go ahead whenever you want.
Thank you, Javier. My job is actually I'm professor for histology and biology, but on the clinical part I'm clinical pathologist in a part of gastrointestinal pathology and neuropathology. That's my segment of the analysis, and in my academy part, I have very long time start collaboration with the pharmacologist and where we do on a lot of animal model. Actually, one of is the induction of the hepatotoxicity and using the acetaminophen usually, and some hepatoprotective drugs which we actually use in analysis. So I will try to make some introduction about that, some introduction about the histology, some important pathohistological pattern which we need to know to analyze the histology slides, and finally some details about the animal model of DILI and some guidelines which we usually use in analysis of our slides. So the first slide, first of all I will tell which we actually anybody know what is the DILI. It represents some acute or the chronic liver disease caused by some medication, and it's well known that the liver actually is the main gland in the biggest gland in our body, where we have the transformation and purification of actually a lot of form of chemicals. Sometimes some drugs in overdose like paracetamol or acetaminophen can cause some hepatotoxic effects. Sometimes some therapeutic dose of some drugs can cause some damage, like some anesthetics. Very rare, but still we have presence of some hepatotoxic agents like carbon tetrachloride, usually in laboratory can cause the damage of the liver, etc., etc. So we can see that we have multiple drugs or some chemicals which can damage the liver, and in this damage we have some degree. We will see how it look like. It's sometimes this is just some biochemical findings, but sometimes we can see the morphological changes. So as anybody know, we usually use this website, which is open access database, LiverTox, where we can find usually updated information about a lot of drugs which is connected with the hepatotoxicity. And in my institute, we also use this database for the finding some new information about the drugs and some new drugs which can also be connected with the DILI. First of all, when we start with histological analysis and try to make some like we say, translation to the humans, we need to know the specificity of the animals which we use for the experiment. If we are looking the basic histology parts which we can find in the liver, we can find some which we say portal triads, and the main structure like some bile ducts, like some hepatic veins and hepatic arteries. And if we are looking classical lobule, we can see the hepatocytes and in the central vein. So this is the main histology which we can recognize actually in whole species, but some some difference we can find between, for example, experimental model and experimental species like rats, like mouse, according to the human. So this is the basic histology which we can see in liver tissue, but this is actually for the human. We will see how it looks like in the mouse. This is on the larger magnification, and everybody knows that in didactic reason we say that this is the primary and this is the border of the these classical lobules, but sometimes in histology something and some borders we need to imagine that we can recognize some structure. So we will discuss more about these zones, and these zones are actually parts of the classical lobules, which is highly connected with the type of the damage. The central vein and the damage about the central vein are actually connected with the drug intoxication or some ischemic changes which we can expect in the liver. I put it here some table where we can see actually the main difference in histology, which we need to know that we can analyze the histology tissue if we are looking, for example, the mouse. The difference can be or similarity in the humans can be analyzing the location, number of the liver lobes, for example, gallbladder and ligaments, and also some histological details which we will discuss. So if we are looking microscopically a liver, which is very important in analysis of the liver, we can see, for example, that the gallbladder is not present in the rat, which is very interesting when you are trying to make a model, for example, of that, because we cannot expect it in the rats, so we can just use the mouse. If we are looking at the surface of the liver, in the human, we have a number of these ligaments, which is actually not present in the mouse and the rats. The liver is actually completely free inside the abdominal cavity, which is not the same in the humans. And that's the some gross changes and difference which we can find in these three species, for example. When we are looking histologically, it looks that actually they are similar, but we can see that the structure, like primary lobules, are smaller in the rat and the mouse according to the human. Also, some portal parts, which we can see in the humans, are more fibrous, and we have more fibrous structure inside the portal part, which is not actually moment in the mouse or the rat. So here is the slide, which I say that actually the portal part are just composed of this structure, which we say that it is blood vessels. We can see the bile ducts, but in the human we have a lot of this fibrous tissue. So any changes in the moment that we have accumulation of the fibrous tissue here in the portal space in the mouse can show us that something pathological goes in the liver of mouse. So we need to know the difference, which is actually normal, which is not normal, that we can analyze and make some interpretation in the histology analysis. So if we are looking the same, just the hepatocytes, the finding, for example, of binucleated hepatocytes are often in all these species, and we can find actually normally binucleated hepatocyte. If we are looking the glycogen, this glycogen is very abundant and in high presence in the mouse, and it's very important. We will see in the future slides for the reaction in acetaminophen animal model. We have a lower concentration of the glycogen in the rats, and it is not so abundant in the human. Actually, we in human cannot see this basophilic accumulation, which is very often in the mouse. So it's not same, actually, if we are looking the human pathologist. We have some specific in the liver pathology, and now I will say what is the most important for the DILI and what we can see in some of animal models. So as a lot of these animal model, which a lot of these experiments which we perform in, and also in my collaboration with pharmacologists, they done usually and routinely some biochemical clinical pathology analysis, which actually represents the biochemistry. And almost all experiments need to include this biochemical part, and as additional findings, it can be the histology. So some of the main biochemical parameters can be, for example, alanine aminotransferase or aspartate aminotransferase if we are looking some hepatocellular injury. And if we are looking, for example, biliary injury, we will look at the serum bile acids and the total bilirubin, for example. And some guidelines for the interpretation of the clinical studies, they say that we need that alanine transaminase need to be increased about 2 or 4 times in the rats or dogs, and it can be a little bit greater than this in the non-human primates. But I think that these guidelines can be also standardized in some further investigation. Also, this total bilirubin elevation are actually very, how we can say, in high influence or some stages like cholestasis, some sepsis, hemolytic stages, and we need to eliminate all these stages that we can get very precisely interpretation of these biochemical results. I find some publications which show what we need to have in some study that we can say and that we can completely interpret in some, I can say, border some hepatotoxic effects of some drug or some hepatoprotective finding of some drug. So in the liver function, we need to have aspartate transaminase, alanine aminotransferase, alkaline phosphatase, and actually free or the total bilirubin. And the next part needs to be histopathology with the analysis of the liver weights and also the histological findings of some patterns like necrosis, steatosis, fibrosis, inflammation, and the hepatocellular abnormalities. So this part, which included the gross analysis of the liver, are very important in the DILI animal model. Actually, this represents very easy methods, and you just have macroscopic analysis of the liver, but I think that's very important. Sometimes when we have, for example, autopsy of this animal, which is treated with something, when we are looking just microscopically the liver, I can predict actually the level of the hepatocyte destruction, hepatocyte disturbance, and if I saw that the liver is, for example, more yellow, that we can expect more some steatotic changes, or if we can find some liver which is with some reddish region, we can expect more intensive appearance of the necrotic region. So this liver weight or body weight ratio, which we actually use in any other study, are very useful and commonly applied in the toxicological study. Sometimes with the tracking of the liver weight, we can find the effects, for example, of the absence in the treatment effects. Usually, we can quantify sometimes some effects of some hepatotoxins. We can see that these changes are very easily to recognize, but they are not highly specific for some stages. We usually try to make the microscopic picture of any animals which is treated, and that's some data which we collect from each experiment, and sometimes try to make a comparison with the biochemical, microscopic, and histology findings which we can see in our results. So when we are looking this liver which is normal and this below which is actually treated with acetaminophen, we can see on the one look that this liver is not good. We have a little bit enlargements of the liver, and we can see that the color become more reddish. Actually, we have this region of the hyperemia, and I think that it's mostly the region of the necrosis which is expected in this animal model. So this microscopic part is very important. I can say once again, not just this animal model, but also in human pathological disease and routine pathology diagnosis. So the most important pattern in liver damage is the changes in some hepatocytes. And these hepatocytes, if we make some grading or the staging of the changes, we can say that we can expect the vascular or hydropic degeneration. We can find some lipidosis or steatosis changes, and we can find this hepatocellular necrosis, and we can see which type we can recognize. So first level of the changes in the hepatocytes include the vascular parenchymatose or hydropic degeneration. All these terms are the same, and they say that we have the beginning of the changes inside the liver. Sometimes we can find these changes are completely reversible, and after treatment we can, and in some time which is spent, we can actually expect that these hepatocytes are completely become better and they not die in any time. So we have reversible changing inside the liver. We have some just accumulation of the water inside the cytoplasm, but this is not one type of the cell death. The next stage, which is actually more prominent and also depending about the name of the toxins, are accumulation of the fats, triglycerides, or something else, represents the lipidosis or the steatosis. We can recognize the two types. One is when we have the huge lipid droplets, which we talk about the microvesicular, and if we have the smaller vacuole or the foamy cytoplasm, we can talk about the microvesicular steatosis changes. So if we are looking, for example, this histology slide, we can see that actually we have the mix of these two histology pattern, and that actually sometimes depends of intensity of the drugs, but sometimes we can say that it depends of the type of the drugs. So some drugs, like for example non-steroid anti-inflammatory drugs like ketoprofen, can cause the microvesicular changes in the hepatocytes, and on the other side we have acetaminophen, which usually cause the microvesicular changes. So according to the intensity drug level, we can expect different type of and the different we can see degree of the accumulation of the, for example, in these triglycerides inside the cytoplasm. The irreversible changes inside the liver is the necrotic changes, and these necrotic changes, I put it here in some table, can be classified inside the apoptotic changes. So it can be focal or spotty necrotic changes, it can be zonal necrotic changes, and also confluent necrosis. So this apoptotic, we can see, we usually expect in some hepatitis, which is hepatitis, or which is connected to the viral, autoimmune, or drug-induced apoptotic changes. These focal are also connected with the same, but the zonal necrosis and the confluent necrosis, we can see we are usually connecting to the some type of ischemia or drug-induced changes inside the liver. So the histology findings in the apoptotic hepatocytes, we usually know as a apoptotic body, acidophil body, or we say consmal bodies, which is usually found in some hepatitis or viral infection, but also we can find it as a rare occasional findings in drug-induced liver injury models. Spotty or the focal necrosis actually represents the small clusters of the hepatocytes or some larger group of the hepatocytes. And in this histology pattern, we usually see the very intensive inflammatory reaction, like here in this picture, which is usually composed of the inflammatory infiltrates, composed of plasmacytes, lymphocytes, or some, which we can see here, eosinophilic cells. And it's not necessary that we can see, mostly if it is very small cluster of the hepatocyte, that we can see this dead hepatocyte. We actually find the reaction of the organism to these dead hepatocytes. In hepatitis, we usually say that it is viral hepatitis, but also in drug-induced hepatitis can be like in viral form, which is actually similar to the viral form, and we need to some immunohistochemical confirmation that it is not viral. Some drugs like halotan, isoniazid can cause this histological pattern of this hepatitis, but it's not so easy to find this and put this diagnosis that it is actually drug-induced hepatitis with some other, which is not completely connected. Also, the focal or non-specific hepatitis actually represents the small focus of the necrotic hepatocytes, which is accompanied with the lymphocyte infiltration. We can see in using of the aspirin, and the chronic hepatitis can be caused by diclofenac and methyl-dopa. All these changes we can find in the liver, but I didn't find any animal model which is actually highly connected with these drugs, that they are using them to produce this animal model. Zonal or confluent necrosis are the most prominent finding, which we find in, for example, animal model which use acetaminophen in inducing the hepatic damage. Centrilobular, which is actually represents the acinar zone 3 necrosis, are usually dedicated to the ischemic changes or the drug-related injury. We can find here, like on this picture, we can see this central part, or the vena centralis, and according around this vein, we can see the dead hepatocytes. If we see this histological picture, that we can say that it is centrilobular necrosis. Here on the corner, we can see the portal space, which is actually not affected with this damage. These damage are highly connected with the actually passing of the drugs, because the highest concentration are here, and we are expecting that these hepatocytes, according to the histology or cytoarchitectonical structure of the liver, is first damaged. Bridging necrosis means that we have connection between these two central vein, or these two necroses, and piecemeal necrosis means that we have periportal hepatocyte. This is this hepatocyte at the peripheria can also be damaged. Confluent necrosis, which can be found in also the same animal model, depends actually on the dose sometimes, or the time of duration of this hepatotoxicity. Cholestasis as an animal model, I didn't find it, but I will show here that these histological changes usually become when we have some impairment of the bile flow, and it can be very mild without any parenchymal inflammation. We can see the inflammation in some cholestatic hepatitis, and we can find also some progressive destruction in small bile ducts in some using of some drugs, but it's not so often in animal model which we can find. Also, granuloma are connected with some using of some drugs. Finding we can see, for example, in some allopurinol treatment or phenytoin treatment, but they are usually connected with some systematic vasculitis or hypersensitivity. And according to this animal model, we can expect these histological changes like formation of the granuloma. Some drugs or some toxins are well known to produce some cytological alteration and changing inside the hepatocyte, like vinyl chloride, combined oral contraceptives or something else, can create some focus of damaged hepatocyte, which can be introduction of the developing of the hepatocellular carcinoma, some sarcomas like angiosarcoma or some adenoma. All these animal models can be interesting in oncology, for example, and using of drugs in modeling of some cancers of, for example, liver. So I will just make a very short introduction or presentation about the basic animal model which we use in my laboratory. And first of all, I will say that the DILI animal model can also be separated into the intrinsic and idiosyncratic DILI. The intrinsic DILI are actually dose dependent and they are predictable. We use some toxins, we use some drug to create some liver damage, and it's not so dependent to some biology of the animal or the difference between the animals. On the other side, we have idiosyncratic DILI, which is actually non-dose dependent, and it is unpredictable. These animal models are well developed, and they are not so predictable like intrinsic, which we use in our laboratory. These idiosyncratic DILI are usually genetic, need some genetic variation, and we will see what can be also be some introduction between before we start to investigate some potential DILI effect of some drugs. So this is the list of the most important, or how we can say most frequent animal model, which is intrinsic type. And actually, if we are looking here, we can see the technique is relatively simple. We just give the animals some drugs, and we can expect that these drugs cause damage of the liver. These two, first the drugs and second is the toxins, are most useful, used in the laboratories. And in my laboratory, we use this acetaminophen. And in the past, we also used this carbon tetrachloride, which we try to escape in now, because we know that's very toxic, and it's not so predictable, and we have very high damage of the liver, which is not sometimes useful for the interpretation of the results. Idiosyncratic DILI, as we say, they have a little bit complicated technical challenges, and they need that we have pre-treatment, which can sometimes include genetic alteration or some immunosuppression or some induction of the inflammation of the animals. And after that, we have application of some drugs, which is interesting to analyze the reaction of the animal model. So the acetaminophen is one of the most common drugs with antipyretic and analgesic effects, and we have well explained biochemical or pathobiochemical mechanism how we get these hepatotoxic effects. Actually, they use the cytochrome P45 isoform to metabolize, and we get this NPQ as a result. And we know that the glutathione is the crucial for the further detoxification of NPQ, for the elimination. And as a result of the overdose of the APAP, we have as a result accumulation of this conjugate of the GSH with NPQ, and as a result we have some oxidative stress and centrilobular necrosis as a result. So here is the histology pattern of centrilobular necrosis, and it is always highly connected with some biochemical changes which we can see in the animals. When we are trying to choose the animal which is most important for the analysis and for the animal model, we need to know that even the mouse and rats belongs to rodents. They are the biochemistry of the APAP, it's not same. And just in mice, we can expect these cell necrosis effects, but in rats, it's not the case. And we don't have cell death in the same condition like in the mice. On the other side, in the humans, it's confirmed that it's confirmed actually in the primary human hepatocyte and also the primary mouse hepatocyte. We can find the same biochemical changes in the hepatocyte, and finally the cell necrosis. A lot of laboratory, and also my laboratory, we use the hepatoma cell lines as a model of the hepatotoxicity and hepatotoxicity in vitro model sometimes, but we need to know that the finally changes in the hepatocytes are not necrotic, they are actually apoptotic. So the pathomechanism here, it's not the same, and we need to know it because the interpretation of the results can be actually different if we make some correlation with the human, for example, in the future. One very important detail is the feeding of the animals, and actually if the animal are fasted before the treatment of the acetaminophen, for example, we need very lower level of the concentration that we can get the same histological changes which we can find in the higher concentration, but the level, but with the animals which was feed between or before the treatment. So this is the moment which is very important when we are creating this model. During the time as a histologist in a collaboration with pharmacology, we try actually each time to make some to find some correlation between the biochemistry and the histology, and that's not so simple. And sometimes we need to have some more detailed histology analysis and more, how we can say, measured details inside the tissue that we can make some comparison. In next few, I can say 5 years, we use some scoring system that we can analyze, or according also to the animal model, scoring system that we can quantify the changes inside the liver, inside the tissue section, that we can say that these animals have this degree of the liver changes, and it can be actually sometimes in correlation with the biochemistry. So we usually analyze, this is some model which we, some scoring table which we usually use, which is actually for the non-alcoholic fatty liver disease. We analyze the steatosis grade, which is very important in acetaminophen model. It can be 0, 1, 2, 4, according to the percentage of the hepatocyte which is affected with the steatosis. Second is the lobular inflammation, which is sometimes very moderate, but sometimes is absent. Is also depends of the time, depending in the development of the some experiment. If we have the experiment which is in acute form, that we cannot expect that the inflammatory response can be very huge. But if we have some chronic experiment, that we can expect that we have more inflammatory reaction to the damage, and as a result we will have, for example, lobular inflammation higher. Balloonization is that part which we talk about, the parenchymatous degeneration, and the fibrosis are all usually found in the later form of regeneration of the liver, and it's also in some chronic type of experiment we usually evaluate. If we are looking at the cell necrosis, we need also to quantify how much hepatocytes are damaged, and that's very useful because it's not same if we have just 25% of the lobules are necrotic, or we can see that the necrosis are very severe and affect more than, for example, 75% of the lobules. Sometimes it can depend of the dose we can see. Sometimes we can see it depend on the feeding. Also, this quantification of the intensity of the necrosis are very important for the further analysis. So I will show here a few publications which we done in the past and before a few years. That we can see that histology is very useful in the publication. We can see something morphologically, that something is changes, but sometimes it can be just small overview in standard histological staining like hematoxylin eosin, where we can show that in a control group are not changes, in group with some, for example, hepatotoxins, we have changes, and if we put it something, we can see the changes is not so intense. But this level of analysis sometimes is enough, but it's usually not enough in the study. We need also have some in some level of the collaboration or the correlation with the biochemistry, and in this publication can be we find it, but the best results which we find is when we use more diagnostic technique and more how we can say histology methods for analysis. In the last year, we put it more immunohistochemical analysis, and sometimes immunohistochemistry can show us something which is not so obvious on standard hematoxylin eosin staining or some other histochemical staining. And the most, how I can say, most important part for the histology analysis is that we have some standardization of from which region we used and how many slides we analyze. So the using of morphometry inside the histology analysis and also the grading guidelines are the best part for the histology analysis. Last years, to more upgrade the level of histology analysis, we use some tissue microarray blocks. So from each animal, we took about the 5 samples, which is usually about the 2 millimeters. So per one animal, we usually have 3 or 4 samples, and for each whole group, we can actually put it inside one block. On my institute, on the clinics, we have this, we say it is vitreal microscope. Now we have almost every histological laboratory have some scanner for histology where we can digitalize these histology slides, and they are easier to analyze. These digitalized slides can easily, in sometimes free softwares, can be analyzed for all these histological pattern which we say in the last slides about the necrosis, hepatocellular velaminization, steatosis, apoptotic. So all these details can be quantified, and as a result, we can see actually that all these findings are more detailed and more serious in the interpretation. Also, we expect we spread these immunohistochemical markers, which can be also be quantified using this software, where we can actually find how many cells are superoxidismutase positive or COX-2 positive, and all this quantification give us more information which can we can make a correlation with some biochemistry and biochemical findings. So I put it here at the end some my internal guidelines which we start to develop and to increase and make more serious results which we get in analysis is that first of all we need to see which type of hepatotoxins we use, if it is acetaminophen or or carbon tetrachloride or some another type of the drugs, we need to know how they make the hepatotoxic effects actually. Second, we need to know which animal we will use. So we saw the difference between the different rodent types, not just according to the human. We need to know, very important in some model, diet status of the animal. Then we need to know how long we will have the daily animal model, if it is acute, subchronic, or chronic model. And according to that, we will have the different histological interpretation or the different histological pattern we will find in analysis. Then we will have the number of the animals. Also, we use about the 7 or 8 animals per group. Lower level, lower number of the animals sometimes can be not enough for the appropriate statistic analysis after that. But with this sampling of the tissue section, we can increase actually the coherent and the actually real findings of the results. Then the gross analysis are very important, and with routine microscopic picturing, for example, of the liver, not just making the weight, measuring the weight, but also the picturing are very important, and we try to put it in a routine exam or laboratory procedure. Also, the standardization in histology procedure are very important, because fixation of the liver, which is longer than 24 hours, can be very I can say harmful for the hepatocytes, because in the period about the 2 or 3 days are the limits that we can start with the unbending of the slides, because the hepatocytes, because actually in the mouse are more frequented. We have a lot of glycogen, and this glycogen will change the inside characteristic of the hepatocyte. So in the first 24 hour, after 24 hour, the fixation and the procedure for the unbending are the best for the results, and also the hydration, which is actually important, and also for the morphometric analysis. In the using of tissue microarray block are very useful, and we can also rationalize the using of immunohistochemistry, and we can standardize of analysis. After that, we can make a digitalization of that and statistical analysis, and finally the using the computer software in the quantification. So this is to my colleague from the pharmacology. We start as we was the students before, like I say, about 10 years ago. Now we are trying to every day increase and to like I say, update our analysis, and I think that we are just on the good way, but we need a lot of enthusiasm for that. So this is the Novi Sad, and I would like to host you if you anytime came to Serbia. Thank you.
Thank you, Ivan, for this detailed presentation about the histopathology. Is there any comment in the forum? Or maybe I can start. So when you presented the data with acetaminophen in preclinical models, like with mice or rats, or in this one of these slides that you showed, what is the time point, the best time point to understand necrosis? Is it like at the beginning, like very early damage, 2 hours, 6 hours after you challenge the mice or the rodent with a PAP, or you have to wait 24 hours to assess necrosis and see all the neutrophils trying to phagocytize all the dead cells?
We usually find after 24 hours, we find necrotic changes, but after 2 days, we need to to wait about 2 or 3 days that we can see inflammatory reaction, because the clearing of the necrotic region is not start at the beginning. And first of all, we have actually that this necrotic region, some of these necrotic cells go to circulation, and you know that we have increase of these enzymes, like in the model of the pancreas destruction with alloxan. In first 24 hours we have destruction of the isolates, and after 2 days we have actually then inflammatory reaction. But here in liver, after 2 or 3 days we can expect, and it increased during the time.
Okay.
And it's also some of our model, some of our experiment actually are hepatoprotective, because we have the pre-treatment of some hepatoprotective drugs or some supplements, and after that we perform the acetaminophen, and after 24 hours we killed the animals and euthanized, and then we have usually acute model of drugs. So if you need some chronic analysis, then we need to wait about 7 days or 24 or for example 14 days, that we can expect that we have more inflammatory reaction and respiratory
But do you see any changes in the immune populations? So do you have any differences in myeloid or immune cells depending on the time point?
Yes.
For example, I mean, the beginning is
yes.
Sorry.
You have this acute immune response, which is neutrophils, and then during the time they will change with the lymphocytes, which we can expect in the chronic phases. So in the first days, they will macrophages activated Kupffer cells. They will try to eliminate these necrotic parts. So Kupffer cells, we can expect that they will be increased in this around the region of the necrotic changes.
Okay, that's interesting. Another question that I had is, apart from necrosis or apoptosis, have you checked any other type of cell death, like this emerging new cell death types, like ferroptosis? Have you measured GPX4 or other markers?
Yes, this we can use some tunnel staining for the apoptosis, and it's very useful. And it's we perform it in the model of in vitro model, usually using these hepatoma cell lines, and the tunnel catch these cells that they are actually in the apoptosis. In the in vivo model, we can find some apoptotic cells, and that's the normal finding, which is in the liver, but it predominates the necrotic type of death of these cells. So in vivo model, we usually don't use this apoptotic marker, because the finding of this apoptotic marker usually didn't correlate with the degree of the necrotic level in this model.
Okay. Any other question? Yeah, Raul, please.
Thank you, Ivan. Very nice overview on the histopathology of the drug-induced liver injury, actually, in animals. I am curious about one of your slides that show the effect, for example, of some drugs in animals. And one drug is furosemide, which in mouse can induce necrosis. I think is necrosis, the lesion. My question is, what is the reason for this? Is because furosemide at high dose has an intrinsic hepatotoxic potential, or there is species differences that may allow for an injury in mouse because of an enzyme deficiency or something like this, or what is maybe the reason?
Just to try to find where I put it.
There was a table you show.
It was in the table. Okay.
In a table.
I'll put it. It's here, and I think
maybe no. Go.
Maybe it's this here.
There is in another table you show several drugs that are able to induce hepatotoxicity in animals and different species. But one of them was carbon tetrachloride and acetaminophen, but also furosemide.
We didn't use I find in literature, so I try to put as an example which drugs can be done it. So hepatotoxicity, but I'm not sure that I.
The table is you can show you can find it if you go on.
Just a moment.
Yes.
Here. Okay. Sorry. So they say that it's in the mice and the limit of clinical relevance, potential interface with metabolism. I find it in the literature that they use furosemide, but it's not so frequent like in some acetaminophen as a animal model for the.
Yeah. It's but the other all the other compounds are typically hepatotoxic, well-known hepatotoxic agent, but not furosemide in humans. And my question is, maybe is the dose that is very high? Can this drug can have intrinsic hepatotoxic potential when used at a very high dose, or is an idiosyncratic species depending idiosyncratic response? Or? Maybe
that's the reason why they use it in the animal model. They put it here at the list that can be potential drugs for the induce of this intrinsic DILI. This carbon tetrachloride we used in the past, but it's not easy to, how I can say, titration of the level which we can use. It's the damage of the liver is very high, and we cannot predict that the liver can be so we have so high damage that we can recognize actually the some hepatoprotective, for example, effects of some drugs. And this other type of these toxins or drugs we didn't use in our investigation. So I just concentrate to these two on the table. My apology about that, but we can use it and try to analyze to see. I was more concentrated to start to standardization of some of model. So I think that if I have standardized some model, that it's useful for the analysis.
Okay, thank you.
So that's also the problem, because sometimes we have not clear correlation between histology and between the biochemistry, and then I usually try to say my pharmacologist, you can give me just the number of the animals and I will give you the grade of the damage, and then they give me the groups, because then they give me the groups that it's not so objective in analysis. So I think that it's good analysis for that, because then the results are actually like they are. So So no, it's not sometimes 1 + 1, it's 2. So you know, in the practical part.
Okay.
And
Thank
we
you.
need to be honest, which is results, not to be linear results, which is actually, I find also in some results of some study, they just put it that it is actually linear. If we have increase of something, we have linear damage. It's not same. It's not also. So I think that we need to be honest in representing of the results.
Okay, thank you.
Okay, thank you.
Hello, Javier. May I say?
Yes, of course, Maribel.
Yes. Thank you so much, Ivan, for this very elegant presentation that you have provided us this afternoon. Indeed, we have learned a lot. I would like to take the opportunity to tell all of you that are attending that this young conference series is that it has been just recently, very recently, published online, a paper that has been that represents the development of a new causality assessment scale. Is a revised electronic version of RUCAN scale, and is named RECAN, that stands for Revised Electronic Causality Assessment Method, and has been the fruit of a international collaboration between the DILIN and the Spanish DILI registry. And it's an electronic version. So if you go into the paper that has been published in Hepatology this month, so you could find that the link to the electronic version. But the news about this new scale is that incorporates liver biopsy findings into the different items that are analyzed and scored to get a final to get the probability of DILI. So liver biopsy, because it's not really very much specific, no, of DILI. We have no biopsy has been done. In the drop-down menu, we have no biopsy down. Non-diagnostic can be suggestive of DILI, but not diagnostic. Or biopsy carries features consistent with a specific DILI. And the other item is diagnostic of non-DILI diagnosis. For example, ischemic injury or alcoholic hepatitis or infiltrating cancer. So I would like to focus on the biopsy carries features consistent with a specific DILI. Do you think it would be possible, because you have shown us that at least in 50 with 50 drugs, we could find granulomas. Do you think it would be possible to get a list of such a specific features that could really give us a hint of DILI?
So maybe it can be some analysis. It can be some. We don't have so free. Now I suppose that you are asking me, Isabel, about the biopsy of liver, which we have in routine clinical
Yes.
practice. Yes.
Yes.
We don't have so frequent. So we at 1 month we have about I think 5 biopsy of liver, and it's not always. It's sometimes connected to some tumor or something like that. But if I go to the past and make some retrospective analysis, and according to the data about the patients, maybe we can make some correlation about it's very complicated in a clinical condition of this patient that we can make some connection for that, because sometimes we don't have enough information about this patient, what he took in which dose he actually into this moment. I'm not sure, but it can be done. It can be done in some retrospective, for example, analysis. I can took past 5 years, for example, and we can get some, I think, date about, I don't know, 500 patients. And according to the history of these patients, maybe we can make some correlation about the drugs which they use and the findings which we can find. But usually changes in the liver are highly connected with the age of patients, some more comorbidity, which can be interfered with this histology which we can find. So this, for example, steatosis is common findings which we can find during the time in the liver. So almost all people, men from after 15, they have some level of steatosis of the liver. Actually, they took some low level of alcohol, it's enough. But, so we cannot easily connect it, but I think that if we eliminated some conditions, that we can make some prediction that it can be connected. Especially interesting for this granuloma. I think that it's case report findings, and because of that, they make decision that this granuloma is connected with these drugs. So I think that it's more case reports, and that they put it in some base that, for example, these drugs can make granuloma in the liver.
Yeah.
So
yeah.
If you want, we can start some. I have in my Institute for Pathology, we have this patient, but it's not too much.
Yeah.
And I need to exclude all them which is from hepatitis, which is diagnosed as a viral, and exclude which is most of biopsy, and exclude them which is for some tumors, for example, biopsy. So I'm not sure how many patients are clearly make a biopsy just for some drug hepatotoxicity, because it's not a clear indication that they need biopsy liver. They done a biochemistry, they say, okay, you have damaged liver, and we will follow up you. Yeah.
Okay.
I have a comment.
Okay.
Can you hear me?
Yes.
Perfect.
Okay. I think that liver biopsy is still alive because in checkpoint inhibitors induce hepatotoxicity, you can see granulomas. It's a very special kind of granuloma called ring granulomas and endothelitis. I think that is a very, very helpful tool now for diagnosis in checkpoint inhibitors induced hepatotoxicity. I have a question. Why did you put tamoxifen as a separate or hormonal drug? Tamoxifen, like methotrexate, belong to the same group of drugs inducing macrovesicular steatosis. Can you have an answer for this issue?
I'm trying to find this slide.
Because one more.
Here.
No, here.
Here.
Macrovesicular is you put methotrexate, but tamoxifen is in this same group. one is an oncological drugs and the other is an hormonal drug, but the couple drug induce macrovesicular steatosis, you know?
I know, maybe I now I saw it. It's not connected, and not
Okay,
maybe it's some mistake about that.
no problem.
Okay, my apology.
I think, Ivan, that you have talked about digitalization. I think digitalization can be a very important tool that lead us to standardization, no? Because what we need when there is a liver biopsy is that different pathologies can read what they see, but and so come up with um, well, an idea of what is really. I'm thinking, for example, about autoimmune hepatitis B and autoimmune versus autoimmune. There was a paper, we could not reach an unanimous diagnosis among 4 pathologists. So, do you think digitalization could give us, could serve us to standardize what we are seeing in the liver biopsy and to quantify also what we are seeing there, no? And help in that way to centralize because you can exchange those images with other laboratories, no? To exchange the images that can be read by different services. What do you think?
Yes, I think that it's future. Actually, we digitalize complete study part in my faculty for histology and pathologists, and our students they learn on digitalized slides. Actually, it was very useful in this time of coronavirus because they can analyze from each cell phone the slides. And this digitalization in pathology, this actually means that you didn't need to sit on your work. You can work on the home. It's very useful, and it's very useful that we can share the slides, and a different center can easily share the slides, and they can make more standardization in the analysis. And I think that it's start before. I'm too young to say that it's a few decades, but I think in the next 10 years it will become routine. And this machine which digitalized slides are more cheaper and cheaper, and now we can buy it in very high resolution, which are very fast and make smallest data file for not so much money, and it's cheaper that pathology sit there. So I find this useful also for the experimental part, because we can first of all analyze these slides in some program. We have a lot of program, which is some of them we can buy it, some of them are free, and as I say, we have different grade and different histological pattern. On this slide, we can just put it and we can number, or we can make some number of this lesion. So we can say that the intensity of necrosis are that percentage. We have some reversible stages, which is, for example, 25%, and irreversible we can find it's not 25, it can be, for example, 45. So we can get some number, and that's important in histology, not just qualitative analysis of the slides, but also the real number that we can compare in animals. So I think that's it's not future, it's today. So digitalization, that's good, very good.
Yes. Thank you so much, Ivan.
Welcome.
Yeah, thank you, Ivan. Thank you for the presentation.
Ivan, one just small question. Do you know other members from Cause Action that are also pathologists?
I'm not sure because I didn't find anyone.
Yeah.
No, my colleague from Belgrade, but they are not. They are from some institute for food, and I'm not sure that I find anyone. Maybe I will try to look at, but it's everybody's pharmacologist.
No, but no, a pathologist, but
I find that it's very important for me, this
yes.
first action, because of that.
Yeah, because it will be very interesting to
I've
have.
worked a lot with pharmacologists, and we actually done all of that. So now we are trying to spread this for also genetical part and some Western part, so we can just more technique for the same results. So
We'll try to see if you could create some, even a small network of pathologists that could give us input on liver biopsy findings, and also to define, as I said to you, the list of specific features diagnostic of DILI for specific drugs. No? Well, it has been very interesting talk. Thank you.
Okay, thank you. So we have the next meeting will be the 16th of February. This is Wednesday at the same time, 4:30. And we will have a young postdoc from the group of Christophe Chesney from BioPredict. And she will present us many hepatoma cell lines. Also, well, some more or less what you showed today, Ivan, but they're going to show us the tools that we have from a company, no, to address DILI, from cell lines and some other cholestatic models, cholestatic DILI models. So it will be quite interesting. So I hope to see you there next month.
Okay. Thank you.
Thank you.
Good to see you all.
Thank you.
Thank you.
Thank you. Bye-bye.
Bye. Bye-bye.
Bye.
Bye.